I haven't seen Johann yet, so I will try to take the moderating chair, which is very easy for me with Mike as the next presenter. We've spent some time together when Mike one time flew to Frankfurt instead of Stuttgart, and we spent a few hours together. I'm very, very pleased that Mike is actually here in Niedernhausen today, not somewhere in Stuttgart or somewhere else. So it's a great pleasure to have him here. Mike Lawlor will talk to us about tissue banks. He will explain why it is important to have these in place and why we, as family members of patients, have an important role to play.
Thank you very much for the opportunity to speak. As I think Holger might have just said, I'm a neuropathologist from Milwaukee and a king among men. I didn't get much else out of that. So I have a sort of a different sort of talk today than I usually give. I'm a neuropathologist. I do clinical work on muscle biopsies. I run a couple of clinical diagnostic labs and a research lab that focuses on Nemelin myopathy and myotubular myopathy. Some studies that my lab performs, I'm the principal investigator, and sometimes I'm just the study pathologist and wedding photographer for other people's studies, including a number of people in this room. But today we're going to talk about tissue repositories, and I want to make it clear up front that I'm not specifically advocating the tissue repository that I run. I'm just talking about tissue repositories in general as a means of supporting research and translation of findings from the mouse or dog phase to the human phase of studies. So the first part of my talk, I'm going to talk about tissue repositories. Now, first of all, what is a tissue repository? So a tissue repository or a tissue bank is essentially the same thing. This is a centralized location where you can store patient tissue and where you can distribute patient tissue. And there's a couple of advantages to that to investigators, but the main advantage to investigators is it makes samples on these rare diseases easier to find, and really the critical thing is it dramatically decreases the complexity of getting approval to do the work. Okay, so as we'll talk about consent, obtaining appropriate consent from patients or patient families for the research use of clinically derived tissues can be a pretty complex thing for an investigator to do. And if you're an investigator that focuses on, say, treating mouse models of disease, you may not really have experience in obtaining approvals like that. And so by having a centralized tissue repository, somebody like me who has to kind of have a foot in the clinical world as well as the research world can undergo most of the regulatory issues on the behalf of the community and then have a very straightforward administrative mechanism for people to get the de-identified tissue samples for their research. And tissue repositories can exist for a variety of purposes. The purposes of the ones that I'm talking about today are just for research. This would be taking clinical tissue and then donating it for research. Now, as far as the process of consenting for a tissue repository, what's involved? So the patient or the family of the patient needs to fill out a consent form. You should be filling out a consent form in a language that you are comfortable with. So our tissue repository has German, French, soon to be Spanish, and English versions of its consent. You can consent for the donation of old cases, say the diagnostic muscle biopsies for your child. You can consent with the intention of donating future cases. So you can say, okay, if I ever have another surgery, I would like to donate that after the diagnostic testing is done. And then once you're consented, you can actually decide whether or not you want to opt out. So at that point, you can say, okay, I no longer want to be part of this repository for one reason or another, and contact the bank. And then in some cases, that leads to just the tissue being unavailable for anything. And in other cases, I know our repository attempts to be able to make that available to the patient's institution for clinical testing once again, if it turns out that they need to use it again. But because we can't predict when people will want something in the future, if a specimen has been entirely donated, obviously we're not capable of going back in time and getting something back or stopping the use of things that have already been distributed. Now, I touched on the advantages of a repository, so I'll skip over most of this. Really, for the purposes of the rare disease community, it's so that there are a couple of places in a given country or region where investigators will know that they can go for access to these rare disease tissues, and it makes it much easier on the administrative side. They can just say to their institution that consent has already been obtained for research. And the third thing, and I don't know how this is done in Europe, but in the United States there is a finite amount of time that your muscle biopsy or other clinically derived tissues should be kept. And beyond that point, there's no requirement for them to keep it. And that's a couple of years, okay? And so in some cases, your diagnostic specimens after several years can get discarded in the name of freezer space, for instance. And so this has certainly happened to a fair number of people in the United States that have tried to donate their tissues, that we've gone back, tried to get the tissues, and then they haven't been there. So putting something in a repository actually makes sure that it ends up getting used. Leaving it in the hospital of origin, if this is a concern for you, you should call the hospital where these things were done and figure out what their policy is. Because your specimens may be safer in a more regulated environment that actually cares specifically about your sample, as opposed to counting on the hospital system to keep it forever. Now, as far as the uses of a repository for an investigator, essentially everything that we've had uses for thus far has been collaborative work or in support of translation into human studies, things like clinical treatment trials. In terms of the collaborative work, we're contacted by investigators. They want something for a specific project. We determine that it's a good idea and not a waste of time or tissue. I don't really care if they waste their time. And then we distribute it. In terms of the translation of animal studies to human work, I'm going to talk more specifically about that in a second. As I'll show you, it helps identify some technical issues, things that may be straightforward in the mouse or the dog, but may be different or less straightforward in humans. And also, it's really important to understand, in the light of all of the wonderful things that we're seeing people present today, transgenic models are pretty genetically homogeneous, okay? So you have a single mutation or a single type of mutation engineered into a single type of mouse, and you are judging your treatment impact on that specific mutation type. Your human patients, as we've seen in some of the wonderful clinical talks here, are very heterogeneous, okay? So you have a lot of different mutations at different spots. You have the background of what the patient has in other genes is very different from person to person. And sometimes that variability has a significant impact on treatment efficacy, on the ability to metabolize things. And you just can't predict how things are going to go when you turn from a genetically homogeneous model to a more heterogeneous thing. And assessing impacts in human tissue allows you to start answering those sorts of questions. So briefly, our tissue repository is called the CMDTR, the Congenital Muscle Disease Tissue Repository. It was founded in 2013 based on the request from a number of nonprofit agencies. Allison Fraze from the Joshua Fraze Foundation was one of the key supporters early on, as well as Cure CMD, a foundation building strength, and where there's a will, there's a cure. And then subsequently, we got more support from Audentes Therapeutics and from patient families. And our goal was to centralize and simplify tissue-related research. Our consenting process I've pretty much described. This is my assistant in the repository, Stacy Cassette. She's the mother of an amylin myopathy patient. You contact Stacy, you tell her she's interested, or sorry, she's interested. You tell her you're interested. And then she will provide you with more information, more consenting documents, and then you can decide if you're interested. And then once you consent, we take care of everything beyond that. We contact your hospital, we get them to release the specimen, we get them to ship the specimen, we pay for the shipment, we bank it, we inventory it, and you don't need to be involved again unless you want to change the status of your donation. We have 101 people enrolled in the last couple of years, more enrolled than donors because not everybody had a tissue specimen. We do have 43 individual donors who have contributed over 260 specimens. Some of the autopsy collections that we had 40 or 50 specimens apiece. And you can see there's 7 CNM patients, 4 of which are MTM patients, and several of the others are related to other congenital myopathies. Our distribution process I've already mentioned, where people approach us, we decide whether or not it's worthwhile, and then we distribute it. And so far, we've had a distribution to my laboratory that I'll talk about, and then a couple of Demelin myopathy groups. And we did return 1 specimen because the patient desired more clinical work to be done. In the next 2 years, our initial goals were essentially to establish a functioning tissue repository, which we did. And our goals in the next couple of years are to increase enrollment and the ability of people to access the data in terms of an online catalog platform so that they understand what we have better. And we have integrated several key leaders from the community in the United States. The people related to centronuclear myopathy are Allison and Sarah here, and they've been wonderful. Okay, so for the second part of my talk, I'd like to talk about how you use some donated human tissue to support translation of research. So as I mentioned, I am a pathologist, which means that I look at slides and I try to diagnose these muscle biopsies in terms of diseases. So that's a pretty specific skill set. As Holger likely mentioned, I am not a doctor of geography, and nor am I the type of doctor you want to run into if you're having a medical emergency on a plane. Which several people know a story about. Now, a couple of people have talked about the pathology of MTM. Really, for the purposes of what we're talking about here, what I want you to focus on is that this is a normal muscle biopsy from a 3-month-old. You can see how big the fibers generally are, and you can see that all these purple areas, which are the nuclei, are all at the periphery of the fibers. In a 3-month-old with MTM, a lot of these nuclei are in the center, like you've seen several times today, and the fibers are too small. There's a number of other problems in MTM, but this is useful in terms of tracking what we're going to talk about. Now, how do we use the human tissue? So first of all, regulatory agencies like the FDA don't necessarily know much about MTM, okay? They don't know much about rare diseases. You have experts there in regulation that are not experts in the specific disease class. So the onus is on the scientific community within MTM to educate the FDA and other regulatory agencies about what actually matters in this disease. And the vast majority of knowledge that has been produced in the last decade on this disease is based in dog and mouse model work. So a group of a number of us in this room put together a review paper recently that described the comparative pathology in muscle between the mouse model, the canine model, and the human model. Well, and the human people, I should say. As you can see here, there are findings that are similar. So you've got nuclei in the center in a number of these things. The fiber size is a little bit messed up, but you also have some differences. So in the human, the organelles tend to clump up. In the canines, they tend to form these ring-like structures. In the mice, it shows something kind of intermediate. Now, these are important things to note because they're probably the same process occurring, but in slightly different ways. And the ability to demonstrate that sort of thing helps us make decisions about what we want to propose to look at in the clinical trial. And it was banked human tissue that allowed us to make this comparison with a fairly large number of patients between my repository and Alan Begg's repository in Boston. In terms of how we translate things, this is an example of pathology from the dog gene therapy study that Anna and Casey are going to talk to you about in a little bit. What I want you to notice is that you've got early stage dogs, so 10 weeks, and then older dogs. And in normal dogs, the fibers just get bigger. And in the diseased dogs, you have small fibers with clumps of organelles, and that gets worse. More fibers get affected. Low-dose animals, you get incomplete resolution of this problem, and in the medium and the high-dose animals, that actually completely went away, despite the fact that it was there before they were treated. Now, in taking this kind of expectation of a pathological finding to the human clinical trial stage, we just need to make sure that the same staining preparations work, some of which did and some of which didn't, and also that we are prepared to collect data in a way that the FDA will appreciate. So, for instance, the way that we took our fiber size information was by scanning the whole slides that were H&E stained and running it through an algorithm that was developed by a friend of mine at the University of Florida named Lin Yang, which recognized all of the edges of the fibers, and his computer program figured out how big they all were. But then we had to manually quantify a number of things, and the FDA is much more comfortable with automatic quantitation. So what we then worked to develop was a more fancy system that allowed us to measure a whole bunch of things at once. So in this example, you can see that you've got some red outlines of the fibers, and then some of the fibers are green and some aren't. So the red outlines are dystrophin staining, which allows the computer to understand where the edges of the fibers are. The green labels all the slow fibers, and then the algorithm measures the fiber size, but also tells whether each fiber is a slow fiber or a fast fiber, in case that matters for the trial. One step further, one of the key indicators in myotubular myopathy is internal nucleation of some sort. So we integrated a green or a blue channel here to those other two and developed an algorithm that automatically on the same scan will determine how many centrally nucleated fibers there are. So when we do the trial, we're able to just run these simple slide scanning things and use computers to generate numbers and thousands of fibers. And my technical staff will not shoot me. Because it would take months and months to do that manually. So in summary, there are a number of repositories available. I would encourage you to talk to people in your area that use repositories or that have them, or that talk to me about consenting for them. They're really a great help for a number of types of tissue related work. The process can be frustrating and confusing if you try it by yourself, but we can definitely help with that. And that this is really critical for the advancement of research. And this is my key lab group that works on MTM-related things. We've got a lot of generous supporters, including a number of people in this room, and a lot of wonderful collaborators, including Anna, Casey, Jim, and people at Dentes. And then just to finish it off, I want to just give you a quick insight on something here. And the fact that I appreciate so much you guys coming to conferences like this, realize that it's a huge burden. So I do muscle disease research because I grew up with a very close friend who died of Duchenne muscular dystrophy when I was in med school. So I know a little bit of what you guys deal with, but I've had the opportunity to spend time with two wonderful families in the last 2 days. Emil's family, as Holger said, is responsible for me actually being here instead of wandering aimlessly around Stuttgart. And Gino's family yesterday, gave me a wonderful day around one of the other local cities, and this is him being cured by some sort of that tasted awful. I wanted to bring this out because this is a difficult kind of talk to give. It's a very your day-to-day lives are extremely demanding. And so this sort of talk is difficult because it's like I'm asking you to do more. And you need to understand how inspiring all of you are just by making your day-to-day happen, okay? And if you can make that continue to happen with these happy kids, then wonderful. And if you're able to consent, at some point, that's wonderful. But certainly, these sorts of messages are not meant to make you feel like you're not doing enough, okay? So, thank you very much.
Thanks, Mike.
Danke, Mike. Gibt es noch Fragen an Mike?
questions? Is there one? Ah, yes.
Habe ich das also richtig verstanden?
So did I get that right? When I sign the consent, or if I signed the consent and there would be a surgery upcoming for our son, in the framework of that surgery, the specimen could be taken? So if surgery would be upcoming for our son, and I had already signed the consent, then during the surgery, the specimen could be taken without further work?
And she has signed the consent before. During the surgery, can they get the tissue that can be part of your repository?
So it's the way my bank is set up, and this is different, I think, in other banks. I only go after the spare tissue that's left over after your diagnostic testing is done. So it would be the same specimen that would be taken for the surgery once they decide that they have as much diagnostic information as they're going to get. Then we would use that consent, a month or two after the surgery to then say, okay, can this be donated, and then coordinate it then. So we wouldn't propose to take additional tissue out of your kids, and we wouldn't propose to interfere with the diagnostic testing as a result of that.
Thank you. My son has a muscle biopsy stored in somewhere in London, one of the hospital.
Okay.
If I have a consent to transfer it over, will it be like small slices being transferred, or the whole tissue will be transferred?
It depends on your preferences and the hospital's preferences. In some cases, they may want to only send slides. In some cases, they will only want to send part of the specimen. In some cases, they may send it all. Now, that would be on the level of things that you wouldn't need to worry about. You would just need to consent, and then we would deal with them in terms of what they are comfortable doing.
Doesn't it make it hard to standardize your work when you have these slices from the different hospitals that are just cut a little bit different all the time?
Oh, it would definitely be preferable to have the actual tissue instead of the slides. But if we ran into a situation where the hospital refused to give us the tissue, and they said they would give us the slides, we'd take the slides. It's better than nothing.
Okay, no more questions. Thanks very much, Mike.
Thank you. I don't need to.